The cell cycle is just the period of the cell's life. So you might know that cells come into existence once one cell divides into two cells, and that one cell, once it exists, is going to be in a period of its life called G1. And G1 is a time when that cell does the jobs of that cell. So it depends on what the cell is. If it's a red blood cell, then it's transporting oxygen compared to if it's a liver cell involved with all kinds of biochemical reactions. But G1 is just when the cell is living its everyday life, so to speak. And once that cell starts to prepare for cell division, what needs to happen is the DNA needs to replicate. And the DNA replicates in S phase. S phase S stands for synthesis, and the only reason for the cell to replicate its DNA is because the cell is going to divide. And if the cell is going to divide, it's going to need two complete, copies of its DNA. So S phase follows G1, and then right before the cell divides, which is called mitosis, it enters into a short period of time called G2 or gap 2, and this is just the final preparations for cell division, and then that cell is going to go into mitosis, or in other words, nuclear division. So everything that we looked at, do you see how they're showing us a green arrow on the outer periphery, and they're calling all of this interphase? Interphase is just the period of time when the cell is not dividing. Okay, so from the point in time where it comes into existence, it's living its everyday life, and then it's going to start making preparations to divide by replicating its DNA in S phase, and then those final preparations, all of this is called interphase. So in other words, interphase equals G1 plus S phase plus G2. Once the cell leaves interphase, it enters into mitosis, and this is when the one nucleus divides into two nuclei. So what I want to show you is the form that the DNA exists in is different during interphase than it is during mitosis. Here's a table from page 12 in our notes. It's basically just a table form of what we just looked at. You can see we're looking at prior to cell division, how the cell is going to be in a state called interphase. And interphase consists of G1, S, and G2. And after G2, the cell is going to divide, and that is overall what people refer to as mitosis. And mitosis has sub-stages, which we're going to go over in a different video. If we take a look at the form that DNA is in each of these states, we see that DNA is in a different form during interphase than it is during mitosis. During interphase, the DNA is in a much more relaxed form, known as chromatin. And it's only when the DNA goes to divide by mitosis that chromatin is going to supercoil into something called a chromosome. Let me show you what I mean. Hopefully you can recognize the ladder structure of DNA, the two sugar phosphate backbones, the bases projecting into the middle between the two strands, creating the rungs, base pairing rules in place. And so we've looked at the structure of DNA, and we know it doesn't stay like a ladder. It actually coils. We would call this a double helix. But then what happens to it, which I haven't talked about yet at all, is the DNA molecule organizes itself. Taking a look at this picture from our book, we can see here's the DNA double helix, and they're showing us that the DNA double helix is wrapping around these proteins that are called histone proteins. And it creates kind of what people refer to as a bead on a string type of arrangement. And so if you see what I'm talking about, notice where the DNA is wrapping around the histone proteins. It creates kind of, what looks like a bead. That's known as a nucleosome. And you can actually see this if you use an electron microscope. And so here's a photograph of chromatin, the microscopic appearance of chromatin, kind of like beads on a string. So anyway, this is what DNA looks like for the most part in a non-dividing nucleus, okay, during interphase. On top of that, what they're showing us here is that this bead on a string type of arrangement actually starts to coil a bit, and this is what people refer to as chromatin. And so when we look in the nucleus of a non-dividing cell, instead of seeing these, tight X like structures known as chromosomes. Instead, it looks, very like thread-like and random, and that's the relaxed form of DNA. That's called chromatin. Once the cell enters mitosis, what's going to happen is the chromatin is going to supercoil into those dense structures that people draw like X's, known as a chromosome. So chromosomes technically only exist during mitosis, even though people use the term chromosome kind of loosely. Technically, there's chromatin during G1, S and G2, and then that chromatin will hypercoil. As you can see here, the chromatin is supercoiling into a chromosome. Let's finish up this discussion of the cell cycle with a consideration of how much time this is taking. So we know that the cell is going to live its life in interphase, G1, S and G2, and when it's done in interphase, which is shaded blue in this picture, the cell is going to move into mitosis, which is shaded purple in this picture. So just in terms of time, let's count backwards from mitosis. We know right before mitosis, the cell is going to go into a phase called G2, and G2 is just the final preparations for mitosis. This is when the cell makes the enzymes necessary for mitosis, make sure all the organelles are there for two cells, and so on. G2 kind of reminds me of the last sweep through the house that you might do before you leave for the airport. Did I unplug the coffee maker? Did I turn off all the lights? Did I bring my ID, my money? Did I lock the door? That kind of thing. It's just a quick, check through the cell, making sure everything's there for mitosis. And G2 only takes about 4 hours. And so right before G2, remember, the cell replicated its DNA, and it replicates its DNA during S phase. And to replicate the DNA, that takes about 8 hours time. And so we're looking at about a half a day here, 12 hours for S phase and G2. And how long does it take for mitotic division? Believe it or not, you're probably looking at only about an hour. And so the whole cell cycle minus G1 is less than 24 hours long. And so if you're looking at a cell that has a longer lifespan, a cell that lives a week, a cell that lives a few months or even a few years, you're looking at that cell spending much of that time, most of that time, in G1. And G1 is when the cell does its everyday job, so it makes sense. So it would spend all that time in G1. That's going to be a variable amount of time, but then when it's ready to divide, then the cell will enter into S phase. There's no reason to replicate the DNA if you're not going to divide. So DNA divides, moves into G2, and then moves into mitosis. And so I just wanted to bring up that point, that G1, the cell could spend a lot of time in G1 compared to S phase, G2, and mitosis.